Hi, exercise 3 deals with the cell anatomy as well as the cell life cycle. First thing to look at is the cell membrane or the plasma membrane. You can use either term. This is the membrane that separates the internal parts of the cell from the external environment. It's going to help control the flow of materials into and out of the cell. It's very specific on what can move in and out. What moves in and out? Why is the specificity there? It's all related to the specific proteins that are embedded within that membrane. That membrane is made up of phospholipids with these proteins that are embedded either throughout the entire width of this membrane, or some of them are just on the surface of it. It's described as a fluid mosaic semi-permeable model. What does this mean? The fluid mosaic just refers to the fact that the proteins are not like embedded in cement. There is some movement, side to side movement. Kind of think of like seaweed flowing side to side. Semi-permeable means some things can move through it, some cannot. And in this diagram, we do see a depiction of, if we can show here, this part here is the membrane itself. These are the phospholipid bilayers because there's 2 layers of them. These are lipids, which means it's hydrophobic. Anything that's hydrophilic, anything that can dissolve in water, is going to have problems moving through this membrane. It's going to be very difficult for that to do. Now, all of these bluish color structures, these are all proteins. So as I just said a bit ago, some of these proteins go the entire width. That's what we call an integral membrane protein. It means it extends the entire width. Some proteins are just going to be on one part of the surface. It doesn't go all the way through and extends out on the surface. Some of these proteins, what we call glycoproteins, have the protein, which would be right here, and then glyco means it has a sugar or a carbohydrate attached to it. Some of them have lipids attached to them. Also, just want to make a point of what's also embedded in the membrane right here. Excuse me, did not want to get that on there. Is the cholesterol. Cholesterol is necessary as part of stabilization for the membrane itself. Passive transport. There's 3 main different types of passive transport: diffusion, osmosis, and facilitated diffusion. Diffusion just means things are going to move across the membrane, going from an area of high concentration to an area of low concentration. That's the way it naturally wants to flow. It does not require any energy to do so. It naturally wants to flow. It's like you have a swift moving stream and things want to naturally flow downstream with the current. That's exactly the same thing that's going on with diffusion. Osmosis deals with the movement of water. So very simply, osmosis is essentially diffusion of water. And then facilitate diffusion, you're still moving from an area of high concentration to a low concentration. The only difference is here you're going to need some help to get across that membrane. You need the help of a protein. And typically, why do you need this help? Because you're a hydrophilic substance. In other words, you are a substance that dissolves in water and it cannot move across the very easily across the hydrophobic lipid layer that's there. So passive transport does not require energy. It's the movement of substances naturally the way they want to flow. Eukaryotic cells, which is what we focus on here, are more complex. They're larger than the prokaryotic cells. So all cells are divided as one of 2 types. They're classified as either prokaryotes, which are very simple. We don't tend to look at those in A&P. Prokaryotes are things such as bacteria. Now, excuse me, eukaryotic cells are going to be things like algae, fungal cells, plant cells, and animal cells. So that's why we look at those. They tend to be larger than the prokaryotes. They tend to be more complex. They do contain organelles or structures inside the cells that are also enclosed within their own separate membranes. So we call them membrane-bound organelles. All cells will have DNA. One big difference between prokaryotes and eukaryotes: eukaryotic cells, that DNA is in the nucleus, so it's separated from the rest of the cell. That is not true in bacteria and other prokaryotic cells. They have DNA, but it's all just thrown in the cell together. These membrane-bound organelles have very specific functions, and they work to help basically make the cell more efficient. Everybody has their own little job to do. So this is a representative drawing of a eukaryotic cell. How do I know it's a eukaryotic cell? Because it does have the nucleus here. Now in the middle right here, it's the wrong color. Let's see if we can do this with the color we can see better. Right here is the nucleus. It has its own membrane. These little squiggly lines in here, that's the DNA that's actually in there. You have other structures such as right here, you've got the Golgi apparatus. That's going to help with the modification of proteins. You're going to have, and back here, the DNA, I was talking about chromatin. That's just another, essentially think of it as another word for DNA. It has to do with the form how the DNA is. Up here, we've got ribosomes. That's a site for protein synthesis. You have the mitochondria. Up here, if I can draw, is the powerhouse of the cell. That's where you're going to make most of the ATP, the energy for the cell. Lysosome, this is the recycling center for the cell. So there's not all of these structures are in all cells, but it's just an example of some of them. So the nucleus, as I said, this is where the DNA is. It's considered a control center for the cell. It is surrounded or protected by the nuclear envelope, has a membrane. That will help to control what can move in and out of the nucleus. Once again, protecting it. So this is just an enlarged picture showing the nucleus. Because it's control center for the cell, it does have to be able to talk to or communicate with the rest of the cell, and that's done by moving substances in and out of these pores that are part of the nuclear membrane. And as the nucleolus is this structure here. It's made up of proteins, and it helps to make ribosomal RNA that's used later. When we look at the cell life cycle, the cell life cycle can be first divided into one of these two different phases. So if you were to think of it as right here, excuse me, that just switched to something else. Interphase and the mitotic phase. Sometimes mitotic phase is referred to as the M phase. Interphase is where the cell is going to spend most of its lifetime. During this phase, as you have a brand new cell, it's immediately in the interphase. It's where the cell's growing. It's where you will eventually replicate or duplicate or make another copy of the DNA prior to cell division. The mitotic phase is when cell division actually occurs. Now we can subdivide interphase. Like I say, the cell, brand new cell, this is where it's going to go, and this is where most of the lifetime will be spent, is in interphase. During interphase, you have a lot of what we call high metabolic activity, and what that means is you have a lot of chemical reactions going on. You're breaking down glucose to get energy. You're breaking down fats. You're making proteins. You're busy doing all kinds of different things. All this at the cellular level. Interphase itself is subdivided into three different parts: the G1, the S, and the G2. The G1 during this phase, this is the first gap phase, the first growth phase, and this is Get this back on red so you can see it better. Like I say, you have a brand new cell. It first enters into the G1. The cell's growing. It's getting larger in size. It's carrying out a lot of different chemical reactions. It's doing its thing. Then it will move into the S phase. The S phase, this stands for synthesis. This is when the DNA replication is going to occur. And then the G2 phase, after DNA replication has finished, this is the second gap. This is where the cell is going to now continue growing, and it is going to finish all the final preparations, getting ready indeed for the eventual cell division that's going to occur. So as I said, this is where it is going to spend most of its lifespan. It is going to be actively growing and replicate that DNA. So as you can see on this drawing right here, you have a brand new cell. And it is not wanting to draw. Here we go. This is all interphase, this gray area. And you can see that tiny little piece of the pie right here, the M. This up here is the mitotic or the M phase. So you can see how much smaller it is compared to the rest of the life cycle. So that mitotic phase or M phase, it can also be subdivided into mitosis and cytokinesis. Technically speaking, mitosis is simply when the chromosomes will divide, and then cytokinesis is when the cytoplasm itself is then going to divide. Stages of mitosis. Now, some books will put this as 4 phases, some will put it as 5 phases. The difference is with prophase, sometimes that is subdivided into prophase and then prometaphase. A lot of places just combine it all into one for prophase. So the stages of mitosis are prophase, metaphase, anaphase, telophase. There's different ways that you can remember this. The way I usually tell students is to remember the order of them is pass me another tequila or PMAT. And so let's see what each of these phases, what's going on, what does it mean? In prophase, what happens here is the chromatin, this is the DNA, it tightly coils. What this means is that the DNA as chromatin, there we go, is usually this kind of strand. Chromosome, same DNA, but when we say it tightly coils up, the same DNA just very tightly coils up like this. So this over here is chromatin, and chromosome is when it is tightly coiled up. So the DNA that's in that nucleus during prophase, that DNA goes from that long stretched out strand to the tightly coiled up chromosome. The nucleolus disappears. The nuclear membrane disappears. The centrosomes that have centrioles inside them, they normally are sitting right next to each other at a 90-degree angle or a right angle. They start to move away from each other. They're going to end up at separate ends of the cell. So they start to move away from each other. And the spindle fibers start to form basically from the centrioles. You see them extending out from it, and they will attach to what's known as the kinetochore. It's a protein that's on the chromosome. So metaphase, think middle. Metaphase, middle. The centrioles by now have moved to opposite ends of the cell, and the chromosomes are going to be all lined up right in the middle. We call it equatorial plate. And we've got pictures to show this in a moment. Anaphase, think apart. Now those chromosomes, that DNA that it already made a copy, remember, of the DNA during the S phase of interphase, but they were connected. The two copies that are connected to each other. They now are separated. Why? Because that's where the spindle fibers are attached, and the spindle fibers start to contract, and so they're going to pull them apart. And then telophase is simply the opposite of prophase. Your nuclear envelope reappears, the nucleolus reappears, those chromosomes that were so tightly coiled up, they start to unravel, or the DNA is not unraveling, but it's stretching out. And then spindle fibers are going to disappear. Cytokinesis, as I said, is technically the division of cytoplasm. It usually occurs so that it ends the same time that telophase is ending. So the two processes, they're technically separate, but they're going to end at the same time. For mitosis, you are going to end up with two genetically identical daughter cells. So those two new cells are genetically identical to each other, and they are genetically identical to what that original parental cell was. And in this diagram or table, has it broken down. Remember, prophase and prometaphase are often combined into one phase, so that would all occur during the same time period, if you will. But at the top, you have the drawings or the depictions of what's occurring with the cell. The DNA, because it's in that tightly tightly coiled form, the DNA is much more obvious than what you would have then at the very end when it's in the chromatin form. You can see the chromosomes starting to line up. That nuclear membrane is gone. Metaphase right here, you can see how the chromosomes are all lined up right down the middle. And then in anaphase right here, you can see how they have been pulled apart, so they're no longer along the middle, so they are pulled apart here. And then in telophase, you're starting to get the beginning of the two new daughter cells. And these down here are actual pictures using an electron microscope showing, as you can see, the highlighted, the nucleus, the spindle fibers are green, nucleus is the purple, or the DNA that's found in the nucleus. So you can see that clumping together. But that is overall the depiction. Make sure that you use your book to understand all of the stages. Can you describe it or draw the various stages of mitosis?